Hello and welcome back to Lesson 2 of Cell Biology. This is on microscopes. And before we begin, I'd just like to remind you to make sure that you have everything ready before you, highlighter, pens, markers, whatever you are using to annotate your booklet. And that being said, make sure you have your booklet in front of you as well. Whether it's a digital copy or a paper copy, doesn't matter, as long as you're making the annotations and making sure that you're doing the knowledge check and the do-nows when we are going through this and any questions that are in the booklet as well. Okay, so that being said, right, so I will let you pause the video right now and attempt these questions right here, 1 to 8. All right, so most of these questions are going to be about the last lesson that we just did on prokaryotes and eukaryotes. So that's why I mentioned, um, if you didn't catch the last lesson, please make sure that you take the time now to go back and make sure you know the answers. So number one says, state which is smaller, a eukaryote or a prokaryote. So this one's going to be a prokaryote. Number two, identify the difference between a eukaryote and a prokaryote. So there's lots of different answers you can have for this, but the most obvious one I went for is in the definition between the two. So eukaryotes have a membrane-bound nucleus, whereas prokaryotes, they don't really have a nucleus. All their genetic material, which is typically inside of a nucleus, is just kind of floating around in their cytoplasm. Um, and so number three asks, state the function of the cytoplasm. All right, this is going to be full of enzymes that help break down anything that is inside the cytoplasm and to speed up those reactions that are inside there as well. Okay. Number four, identify what a cell wall is made of. This one is going to be cellulose. Okay. Number five, state the function of a ribosome. So again, make sure you know the functions of all the organelles that we've learned about so far, and ribosomes are responsible for making the protein in our body, right? So that's going to be a site of protein synthesis. Make sure you try and use the scientific terms, but if you put the place where proteins are made, I would probably accept that. Number six, identify the subcellular component. So whenever you see subcellular component, think of organelles, okay, that a plant and bacteria contain, but an animal does not. So you're going to have to think about this one because we did learn about animal cells, plant cells, and bacterial cells. Okay, the answer to this is cell wall. So again, when you see subcellular, all right, so if it was a cellular component, that would mean at a much bigger level. But since it says subcellular, it means smaller than a cell. And number seven, what is the function of the mitochondria? If you put powerhouse of the cell, I will give you a half mark for that because that doesn't really tell you much about um, what it does. But if you put that it's the site of respiration, or if you put to produce energy for the cell, that is correct. The phrase powerhouse of the cell is more to help you memorize what the mitochondria is known for. And the last one, identify what is found in the nucleus. So this one I mentioned earlier on question 2. The nucleus is responsible for making sure it has all the genetic material inside there. Okay, so only found in eukaryotes. All right. So if you need a little bit of extra time to pause the video and record all these correct answers, please do so. But we're going to move along. Make sure you correct them as we go on. So today in our microscopes lesson, we're going to be making sure that you know the differences in magnification and resolution of light and electron microscopes. So we're going to be learning about those two different types, and we're going to learn how electron microscopy has increased the understanding of organelles. All right, so a lot of this is going to feel, you've probably learned this before because we do touch on this in year 7 and 8. Um, but don't feel bad if you don't know it because there are some things that people remember better than others. So just pay attention and make sure you take it all in, okay? Right. So first off, we're going to talk a little bit about the history behind where the microscope came from. So as you all know, in kind of medieval European times, religion took a very, very heavy had very heavy control over what people could believe, what they could practice, what they thought was the truth at the time. Okay, so I'm just going to read this off in the booklet here on line 2. It says, before the reformation of religion across Europe in the 16th and 17th century, there was very little known about the human body as science was frowned upon. Okay, makes sense. However, with the new freedom from the Catholic Church that the Reformation allowed, a new age of investigation arose. So I want you to go ahead and highlight that section right there because that is kind of the starting point of where microscopes came from. So people like Isaac Newton and Copernicus were kind of the forerunners or the pioneers in discovering a lot about um, the science around us, particularly the world that we cannot see. So that kind of encompasses two different worlds, the world kind of beyond ours in space, and also the world at a microscopic level, which is here with us, but with our naked eye, we cannot see. All right. So moving along, the microscope is very basic form, or in its very basic form, came to prevalence during this time of discovery. So during Isaac Newton's time, during Copernicus's time, we got to learn about microscopes. So no one really knows who was the first idea. There was a lot of speculation about the scientists from different countries that made it, but there is no actual evidence as to who actually made the first microscope. But we do know that a lot of scientists made their own modifications on the microscope to be able to study the things that they're famous for. So Robert Hooke, picture on the right there, is also a very, very famous physicist. And Hooke, he had worked with Robert Boyle during his time working on the ideas of gas. But in the 1660s, Robert Hooke was one of the original founders of the Royal Society. Um, so this guy, Robert Hooke, made the book called Micrographia, right? And he was kind of the first ones to make discoveries about the cell. All right. The only part that I want you to highlight in this section here is this part where it says, however, this idea was improved by several scientists to allow larger magnifications. So the reason why I have you highlight this is because so far we're just learning about the history of the science behind the microscope, which is quite important, but we can't really ask you much more than, who is this person? Why was it so important kind of question. So as long as you understand that um, the microscope was kind of improved on over years and years by different scientists. Um, that is what I want you to know. And like I said, he was the first one to create the word cell, and that's just because of where he was as a person. So they kind of look like monks' cells, excuse me, in monasteries. Okay. If you look at your booklet, you'll find a link to a video on YouTube called The Weird History of the Cell Theory, which I strongly, strongly recommend that you take a look at because it does tell you kind of what I just talked about, the history of the cell, or sorry, the history of how the cell became to be, and it talks a little bit about Robert Hooke and who he was, how he kind of brought about one of the first microscopes to use. Okay, so take a look at that if you have time. Not very long. Right. So the first thing we're going to look at is the light microscope. And normally in school settings, I would have a microscope in the classroom to show you, take a look at what it is. But hopefully you had the chance, either in year 7 or 8, to actually handle a microscope and see how to use it. I know it's not the most exciting thing in the world, but the history behind this and the science that goes into a microscope is actually quite fascinating. So today, the basic light microscope remains almost unchanged in basic principle. So basically, a light microscope has a lens, like a magnifying lens, and an eyepiece to view a specimen, right? So most basic microscopes are compound microscopes. Compound meaning that there are multiple microscopes in that device that you're holding, right, in the microscope, to increase the amount of object that is visible to human sight. So that's we're going to highlight that right there, just to make sure that you understand that a microscope just isn't like a regular magnifying glass that we can buy from the pound shop or something like that. It is a series of lenses that will increase what you can see in an object. All right. So that itself, that definition right there, is actually called magnification. So when you magnify something, what you're essentially doing is blowing up the image to a proportion or a size that you can actually see very, very small details in. All right. So it doesn't stop right there, though, right? When you have a picture, let's say on a computer, and you want to see the details, you try and enlarge the image, and you'll find out that that image stays very, very grainy, or you can't actually see the details. So what we say to those ones is they have low resolution. Okay, so that brings me to the second point. If you go down the paragraph at the very bottom there, um, we come upon a term called resolution. Okay, so the resolution of the images is how clear they are and how easy it is to define the difference between two points or two parts. Okay, so whenever you look at a picture of a cell or anything like that, most likely when you publish it in a work, it will have something called total magnification. All right. So when we're talking about magnification, we generally have to put it on there because if you have a picture of something, um, and it's kind of hard to tell what it is, it helps to put that magnification so that when scientists look at it, they know, well, this is actually at 400 times magnification, so we know that it has to be extremely small. Okay, so I'm going to go back to the middle of the paragraph where it says to work out the level of magnification of a microscope, you basically multiply the eyepiece magnification by the objective lens. All right, so I'm going to, on the next slide here, I am going to show you the different parts of the microscope, but I want you to understand that when you calculate total magnification, it's actually quite easy. Okay, lots of students have problems with this. So total magnification equals eyepiece magnification, which is generally 10, right? But it's different for every microscope, so double check that it's 10 times, and then the objective lens, which is the one that you kind of spin around. And there's generally going to be three on a light microscope, but there sometimes could be four, and those four generally come in sizes of 4 times, 10 times, 40 times, and 100 times. Okay. At the school, I do believe that most of our microscopes only have three objective lens, 4, 10, and 40. So for instance, if I was looking through the 4 times magnification, I would multiply 10 by 4, which would give me a total magnification of 40 times. All right. So very, very simple, nothing too complicated about that. All you have to do is multiply those two numbers together, and that's what the total magnification you get. So looking at this picture, it might look very similar, might not, but that's why I'm here to kind of go through it. So this actually is a picture of a very primitive light microscope. So um, like I said before in the earlier slide, the basic model of the light microscope hasn't changed too much, but we have with the evolution of technology, we have kind of improved it in slightly, and I'll explain in a little bit once we get to those parts. So I'm going to go in order here with the letters A, B, C, D, E, E, F, G, and if you know some of these parts already, go ahead and label them, but I will be going through them in order. So starting with A, you're going to have two dials on most microscopes, and one dial will be much bigger than the other one, and the bigger one is called the coarse focus. So the coarse focus will actually move the stage up and down, or in this case, it's actually going to move the body tube up and down, and that is kind of a fast way of bringing your specimen into focus, right? And the smaller dial will actually be called the fine focus. So these two dials are used together just to make sure that your specimen is in focus, right? So you use the coarse focus first to get it generally in focus, and then you use the fine focus to kind of do the fine tuning, so get it even more into detail if you need that precise measurement. All right. All right, part C. Now, this is where it gets a little bit different here because the microscopes that we have do not look like this. Okay, so in the picture, in the diagram, you'll notice that it looks a little bit like a mirror. All right, and that's because this is where the name light microscope comes from. So these microscopes are dependent on light coming through the stage, right? So in the early days, these light microscopes only had a mirror to kind of bring in natural light from the sun. Whereas today we can actually put in a light bulb and we can connect it to an outlet and have light come up from a light bulb. So very primitive ones will have mirrors. They're the cheap ones. And obviously you need a well-lit room in order to use those. So that's why we have electric light microscopes today. All right, moving up to part D at the top right there. So this part is the eyepiece. So this is one where I mentioned before. It's usually going to be 10 times magnification. It could be different, but it should say on the microscope itself if it's anything otherwise. But 10 is usually a safe bet. All right, part E is called the body tube. All right. Now, the body tube has been made smaller in recent years, and that's just because we have much better information about how lenses work. So you won't find a very large body tube like this on modern light microscopes, but it is good to know. Okay. And part F is going to be your objective lens. So this one looks like it has 4 objective lens, if I'm not mistaken. But these objective lens, you can spin them around to get to the objective lens that you want to work with. So 4 times, 40, 10 times, 40 times, or 100 times. And generally, the more magnification they are, the longer the objective lens is going to be. And they sometimes are color coded on the microscope itself. So, and you should be able to tell which one's which just by it should be written on there, or there should be some sort of diagram telling you which one is which. And the last one is the one that I keep saying over and over, and that is the stage, right? So in the microscopes that we have at school, your coarse and fine focus will be the one to move the stage up and down, and you can actually see it move up and down if you use the coarse focus. So the stage is where you're going to be able to put the specimen on there. You can see 2 little clips on the stage, and that's going to hold the glass slide in place so it doesn't move around as much. All right. So like I said, you may have already learned the different parts of the microscope already, and you might know many of these parts. And if you don't, please make sure that you have the correct answers on each of the letter and the lines, okay? So that's the light microscope, not much to explain there. So we're going to move on to the next part, which is the electron microscope. So this one is going to be quite a bit of information, but don't worry. As always, I'm not going to get into too much detail about the electron microscope because there really isn't much to talk about it, especially at your level right now. So I'm just going to read it out online too. By the mid 1800s, scientists realized that there was a little more improvement that could be made to a microscope, meaning that magnifying to a higher level was near impossible. However, as technology has advanced, discoveries have aided the cause. In the 1920s, physicists discovered that accelerated electrons behave in a vacuum much like light. Furthermore, it was found that electric and magnetic fields could be used to shape the paths followed by the electrons. So, this section right here is essentially how electron microscopes work. To put it in simple terms, basically what we do is we shoot a bunch of electrons at an object or a specimen, and those electrons will either pass through them or they will bounce off them, depending on what type of electron microscope you're using. And what the electron microscope will do is it will then collect those electrons, and you will be able to see an image on a screen of what you're looking at. So there's no eyepiece lens for an electron microscope, and you have to have it hooked up to a computer and a program that is specifically designed to interpret the collection of electrons. Okay, so some names on there: Ernst Ruska at the University of Berlin, along with Max Knoll. So these two were responsible for creating the first transmission electron microscopes. Short version of that is the TEM. Okay, and Ruska was awarded a Nobel Prize for that, and rightly so because the electron microscope has opened up a whole new world of the cell. So this diagram here is actually in your booklets. So the most important part is it has the same components basically, in that it has the lens, and not just one lens, it actually has a lot of different lens. It has the electron beams, which is where the electron microscope gets its name, and you have the camera at the very bottom, which collects all the electrons that are shot through. Okay, so hopefully this is making sense so far. I know it might be a little bit confusing, but I'm going to try to explain it as much as I can, and I'll show you some images of what electron microscopes can actually show you. Okay? All right, so first thing is kind of reiterating what I just said. The electron microscope creates an image by using a beam of electrons. So Make sure you highlight that. Excuse me. That's going to be your important part there. This leads to higher magnification being used and a greater resolution of images than a light microscope. All right. So some ways that an electron microscope is much better than a light microscope is that you get better magnification, or high magnification, I should say, and you also get to see objects in much finer detail. So high magnification, high resolution. These are two qualities that an electron microscope just can't be beat by with a light microscope. And that being said, there are two types of electron microscopes. So there's even more of a division going on. So you have two types of microscopes, light and electron. And then from electron microscope, you have two more microscopes. You have transmission electron microscopes, and you have scanning electron microscopes. And I'm just going to call them TEM and SEM from here, just because I don't want to say the full name. So TEM involves a high voltage electron beam. So lots of difficult words here, but I'm going to make this as simple as possible. So the basic thing I want you to remember about TEM is that TEM produces a 2D image, black and white. Okay, 2D image. Now you might think, well, most images that we see about cells are 2D, and that's not actually true, because if you look at the SEM, so SEM creates a three-dimensional surface scan, and each one has its cons and its pros. And I'll just tell you the biggest one. So TEM, which produces a 2D image, you're able to see the insides of that cell. Kind of like if you were to slice open like an apple, you'd be able to see the insides of it. Okay. But an SEM, you can only see the surface of a cell. So imagine same thing, an apple that isn't cut. You can't, like, you know there's a seed in there, but you don't actually know it until you open it up. Okay. But from a surface point of view, you can see that it's an apple. Okay, and most of the times, the SEM can produce a color image, but most of these are actually because the scientists, after they take the image, they will add in the color to help differentiate what's what in the picture. Okay, and just to finish it off, that last sentence there, both types can magnify 10 million times more, allowing us to see subcellular components or organelles of bacteria that are only a few micrometers wide. So remember, bacteria are much, much smaller than animal and plant cells. And even with a light microscope, you wouldn't be able to see much about a bacteria. But with electron microscopes, you can see pretty much how they work, how they attach to your cells, things like that. Okay, so let me show you a picture of what an SEM and a TEM look like. Okay, or sorry, what sort of images you'll see. So on the left, you have the SEM, which again produces the 3D image, and then the TEM on the right side, which is the 2D. Okay, now you'll notice that on the SEM, you can see that these are pollen, by the way, from plants. So you'll notice that at the SEM, you can see that they're just kind of like round objects with they look like they're spiky. Okay, so those spikes are actually receptors that will attach itself onto where it's supposed to go in another part of a flower, right? Um, but you don't actually know what parts are inside. Like if I asked you, if you just had the left image, and I asked you what organelles are inside that pollen grain, you wouldn't be able to tell me because you can't actually see inside. Whereas if you had both of them, okay, the TM would be able to tell you, well, I can see that there's a nucleus because there's a small black dot in the center, and it is enclosed in some sort of membrane. And most likely those white dots are going to be either permanent vacuoles or some other organelle that helps it function. Okay. Um, Now, likewise, if you just had the TEM, you wouldn't be able to tell that this pollen grain has a bunch of spikes on it. And if I asked you the question, well, how you know how does a pollen grain attach itself to an egg, or how to get fertilized? And unless you had the image on the left, that way, you could tell me, well, there's actually receptors on the outside of that. So there are good and bad things about both the SEM and the TEM. It really does depend what you're looking for when you're doing this sort of research. Okay, so there you go. Um, about the electron microscope. So make sure that you understand the difference between the light and electron microscope. Maybe you can memorize some of the different components. Definitely memorize the components of a light microscope because that is the easy one, and that is something that we could definitely ask you on an assessment, or they can ask you on the GCSEs. Okay. So at the very end of the lesson, before the knowledge check, you'll see this learning activity 2, where it says, with your teacher's direction on your student's notes page, make a table comparing the main features of light and electron microscopes. So this question is worth 6 points, which means hopefully you're able to tell me 6 distinct points of the advantages and disadvantages of light and electron microscopes. So this is how I would do it. Make a table, 2 columns, one that says light microscope and one that says electron microscope. And then in each column, tell me what are the advantages and disadvantages of light microscopes. And then on the other side, tell me the advantages and disadvantages of electron microscopes. Okay, so go ahead and do that. Pause the video right now and we'll come back and I'll let you know what the answers I got. Right. So, if your answers do not look exactly like this, don't worry because obviously this is where I would ask different students as to what answers they got and write them out on the board. Um, but these are generally the answers that I'd be looking for. So, a light microscope is very cheap and it's easy to use. And that's just because a light microscope doesn't have as many complicated parts as an electron microscope. As we mentioned, electron microscopes they're much larger. They require a computer in order to look at the image, so much cheaper for a light microscope, which is why schools can afford them. One of the disadvantages of a light microscope, as we mentioned before, is you don't get that magnification, you don't get that resolution, so you can't see very small organelles. I'll give you an example right now. So all cells in plants and animals have ribosomes, which are the site of protein synthesis. With a light microscope, you would not be able to see a ribosome. They are too small, too tiny, even though there are lots of them in the cell. Okay? With the electron microscope, I'll start off with the disadvantages because we've already kind of mentioned them. So electron microscopes are very expensive. If you just Google electron microscope price, you'll be able to see what I mean by that. And they actually need an extensive training to use. You can't just go up to and press a button and expect to get an image out of it. You need to know how to use a microscope like that. You need to know what program to use, how to use that program. There is just way too much stuff that you need to know in order to use an electron microscope. Which is another reason why our school will not buy them, even if we had the funds to do them, even if we had the money, then that would require teachers to learn how to use them as well. But let's move on to the advantages. The electron microscope, as we know, is very powerful. So you get high magnification, high resolution. So you are able to see extremely small organelles, such as the ribosomes. Okay. All right. So that's pretty much it for the electron microscopes and the light microscopes. The last thing that you're going to do is finish off with this knowledge check. 10 questions. Pretty simple stuff. Look back at your booklet if you need to, but I'll give you the chance right now to pause the video and we're going to go through this quite fast, okay? Right. So, state the name of the author of Micrographia. So this is, who was the scientist that wrote this book? And that was Robert Hooke, right? Number 2, identify what building block what building block of life he observed and named. And that was obviously what we've been talking about, the cell. All right. The microscope is a series of lenses, right? So not just one lens, not just one magnifying glass. There's actually a series. We call it a compound of lenses. Number 4, define the term magnification. So I just took it straight from the booklet. Increasing the amount that is visible to the human eye. You can't really get more technical than that, just because I understand if you say to make an image larger, so that would work as well. Number 5, define the term resolution. So this one, same thing, I pulled it from the booklet. How clear an image is, or the ability to distinguish between two different points. So think back to an image file on a computer, and if you enlarge that image, is it still blurry, right? If it is, then it has low resolution. But if you're able to, tell apart two separate points on that picture, then it has very good resolution. Number 6, state what an electron microscope uses instead of light. Well, it's in the name, electrons. Number 7, suggest a top magnification for a TEM or a transmission electron microscope, and that is, as we said, 10 million times. Number 8, name the two scientists who patented. Okay, patent just means who had the first rights to make it, who you know made it first, the first TEM, and that was those two scientists, Ernest Ruska, who won a Nobel Prize for that, and Max Knoll. And number 9, using the keywords, write a concise paragraph to summarize this lesson. So obviously your answer is going to look very different from mine, but if you have some of the main points that I wrote down, that's all I ask for. So microscopes help us see another world that we never knew about and also helps us understand how things work at a molecular level. Electron microscopes are much more powerful than light microscopes. I mean, if you had that down, I would give you full marks for that because that does kind of tell you everything that you need to know about microscopes at a general level. Okay? If you gave a little bit more detail as to how each of them work, that's completely fine as well. And number 10, give two advantages for a TEM being used rather than a light microscope. So, I mean, you're making a comparison between an electron microscope and a light microscope. So you can use any 2 points that we mentioned in the previous chart or the table that we just made on the previous slide. So, I mean, high magnification and high resolution. Those are two extremely good points. Actually, I think those are the only 2 points that you can use for using an electron microscope over a light microscope. Okay. All right, and that's pretty much it for today's lesson. So, please make sure that you do have the correct answers. You can just go back if you need to find the parts that you need. All right, and thanks again for watching.